Right, hello. This is the first of 3 lectures to help you pass the genetics questions for the MRCP CH Part 1. In this lecture I'm going to talk about calculating genetic risks, and I would advise you to have a pencil and paper to hand so that you can do any workings out for the calculations, and also so that you can actually commit yourself to the answers to questions. That way it's much more obvious when you've made a mistake and you can then learn from it. In this lecture, what I'm going to do is to give you a general understanding of the principles behind genetic risk calculation, to give you some understanding of some of the principles of probability that we use to calculate genetic risks, and then to look at some examples of calculating clinical risks for a number of scenarios for different forms of inheritance: dominant, recessive, and X-linked. But let's start by thinking about risk and probability. And for this, I'd like you to imagine a clinical scenario. Mr. Blackburn comes for advice about cystic fibrosis. So the first question I'd like you to consider is Mr. Blackburn a CF carrier? Well, the answer clearly is we don't know. I don't know, and you don't know, because we don't have enough information about him to know that. However, there is a question that we can answer sensibly, and that is the question, what is the chance he's a CF carrier? We don't know for certain, but we know that there is a chance. So what is that chance? Well, all we know about Mr. Blackburn is that he's a man. We know he's come to our clinic. So all we know about him is that he is someone from the general population. And his risk would therefore be a general population risk of around 1 in 25, or 4%. So that's all we know about Mr. Blackburn at this point. But we do know something. However, he then tells you that he has some sort of family history of cystic fibrosis. This new information allows us to alter the probability. When he first walked in, we simply knew that he was a human being, presumably from the general population that we deal with. But we now know that he's no longer just a member of that population, but he's a member of a more specific population, that of people who have a family history. So we don't know precisely what his risk is at this stage, but what we do know is it's likely to be greater than the general population risk. So his risk is likely to be greater than 1 in 25, greater than 4%. As the consultation proceeds, Mr. Blackburn tells you more. He tells you that his brother had cystic fibrosis, but that he himself is healthy. Now, we know from our clinical knowledge about CF that this is a childhood onset disease, and if this man has come as an adult and he's healthy, we know that he himself does not have cystic fibrosis. But we do know from what he's told us that his brother had the disease, and that means that both his brother and himself share the same parents who are both carriers of CF. So Mr. Blackburn must have a 2/3 risk, or 67% risk, of himself being a carrier. Of course then, as things happen in the clinic sometimes, Mr. Blackburn pulls out of his pocket the letter from the DNA lab and shows us his positive carrier test result, which shows that he is in fact a CF carrier and a mutation has been identified from his blood sample. And so now we know that the probability of him being a CF carrier is 1. This is no longer a matter of uncertainty. We know for sure because we've seen the blood test result. So, what this illustrates, I hope, is an important point about probability. Probability is putting a number on our uncertainty. It's quantifying our uncertainty. And as you saw from the example, as information comes to light, as new knowledge is revealed, the probability of an event changes. It's not fixed. So although obviously in the exam you're asked to calculate risks and probabilities for a given scenario with a given amount of information, in the real world, probabilities are not fixed. They may change as new information becomes available. So as Mr. Blackburn came into the clinic, we knew nothing about him. We were able to estimate a probability based on simply him being a member of a certain population. By the end of the consultation, our uncertainty had reduced to nothing. We knew for certain, but only because certain information had been brought to light. Now, the other thing which people sometimes have difficulty with probability is how to join probabilities together. Now, of course, there's a whole mathematical theory of probability, which I'm not going to go into because it's not relevant to what you need to know. But there are 2 particular important things that you do need to know about, and that is how to join 2 probabilities. Now, if 2 probabilities are for independent events, events that do not influence each other, then the chance of those two events happening at the same time is the product of the two individual probabilities. You multiply them together. However, if the two events are mutually exclusive, only one or the other can happen, then the probability of one or the other happening is the sum of the two individual probabilities, i.e. you add them together. So, let's take a clinical example to illustrate this point of how you join probabilities together. So imagine that we have a couple who are both CF carriers and are planning their future family. And for their own personal reasons, they've decided that it would be different for them to have one ill child to look after than to have two ill children to look after. And so they would like to know what is the chance of them having two children who are both affected by CF. And this is assuming we don't do any prenatal testing and such like. So the first question we have to ask about this scenario is, are these independent or mutually exclusive risks? And I would suggest you commit yourself now and write that down. What do you think? Well, these are independent risks. The CF status of the first child does not affect the CF status of the second child. These are two separate fertilisation events which don't affect each other. And so this independent pair of risks are multiplied together. Now, because we have two CF carriers, the probability of any one child being affected by CF is 1 in 4, 1/4, and so the probability of them having two affected children is 1 in 4 multiplied by 1 in 4, 1 in 8. But here's a different scenario. Supposing we have two CF carriers again, but this time they want to know what is the chance of them having a child that could pass on the gene down the generations. They want to know whether or not a child might have the gene at all, whether they could be either affected or a carrier, i.e. a child who does not have two normal CFTR genes. Now then, are these two events independent or mutually exclusive? Again, write down what you think. Commit yourself to an answer. Now these are mutually exclusive events. A child can be affected or they can be a carrier, but they can't be both at the same time. So the chance of one or other of these events happening in a single child is the probability of either of them happening added together. So the chance of a child being a carrier is 1 in 2, the chance of being affected is 1 in 4, so the probability of them being either a carrier or affected is 3 in 4. So, genetic risks, typically you're asked about carrier risks, the chance that a child or offspring will be a carrier, and recurrence risk, the chance that in a family where one individual's been affected, there'll be another affected individual. There are sometimes questions in real world about disease instance risk, but these tend not to come up in the exam. And these kind of risks, carrier and recurrence risks, depend primarily on two things. They depend on the mode of inheritance of the condition and the family tree, and of course the position of the individual of interest in that family tree. The disease instance risks tend not to come up because they tend to be based on empirical data rather than on calculated risks. So, let's try a question on genetic risk. Question 1. Which of the following are true regarding CF? Have a look through, and for each question A, B, C, D and E, decide whether you think it's true or false, and I suggest you write the answer down. So let's look at the first one, which is specifically about a risk calculation. Now, is this true or false? An unaffected sibling of a CF child has a 2/3 chance of being a carrier. Well, let's think about what's going on in the family tree. Here's the family tree we're talking about. A child has CF, and they have an unaffected sibling. Now, if the child has CF, that means they must have inherited 2 copies of the CF gene, in this case small c. If the child has inherited 2 copies of the small c gene, that means they must have inherited 1 copy from each of their parents. Therefore, both parents must be carriers of CF. That means that there are 2 ways in which a carrier could result, shown in yellow on the square on the left. Big C, little c, and little c, big C. And as you can see in the square, there are 4 possible outcomes. There are people who have no CF mutation, big C, and people who have 2 CF mutations and are affected, small c. So your initial thought might be that there is a 2 out of 4 chance of the individual being a carrier. However, in fact, because we know the sibling is unaffected, there are in fact not 4 possible outcomes in this case. There are only 3 possible outcomes because we know one of them has been excluded. The small c possibility is not true. because we know the person's unaffected. Therefore, the chance is not 2 out of 4, but 2 out of 3. And so, question 1A is true. So, here's question 1 again. Question 1A is true. Question B is simply a matter of knowledge, and that's false. Question C is a misconception about the use of a sweat test. Sweat test is not a test for carrier status. It's a test for whether a person's affected by CF. And so this is a misconception about the test. Part D is of course true and is commonly used. And part E again is a matter of knowledge, and as it happens, most affected males with CF are sterile. So the answer would be false, because the answer is about 100%. Right, so let's move on to question 2. Now question 2 is a common enough scenario. You're asked to give genetic counselling to a female carrier of an X-linked recessive disorder and her unaffected partner. So which of the following are true? I suggest you write down the answer you think, of which A, B, C, D and E are true and false. Now, let's think about what's going on in this scenario. I've drawn the relationship here and shown you the X and Y chromosomes involved. Now, just for clarity, I've put the X and Y chromosomes from the male and female in different colours, so you can see where the chromosomes are going. The man, as far as we know, is unaffected, and so is a normal X, and, of course, a Y chromosome because he's male. The female, we've told, is a carrier, and so, as with every female, she has 2 X's, but one of them carries the mutation, indicated here with the apostrophe. So they have 4 possible offspring, half of them male and half of them female. For the male offspring, because they're male, they must all inherit their father's Y chromosome. Of course, that's what makes them male. They will then inherit one of the 2 X chromosomes from their mother, either the normal one or the one carrying the mutation. And so half the boys will inherit the normal X chromosome, and half will inherit the chromosome with the mutation on. And because they have no other X chromosome to carry a normal gene, they'll be affected. For the daughters, they must inherit the X chromosome, the blue X chromosome, from their father, because that's what makes them daughters. But similarly to the boys, they will inherit one or other of the 2 X chromosomes from their mother, either the normal one or the one carrying mutation. But because these girls are female and this is an X-linked recessive disorder, the normal X chromosome, the blue X chromosome from their father, is able to mask the effect, the clinical effects on the mutated chromosome from the mother. And so these girls are carriers rather than affected individuals. And so using this information we can then look back at the question and give the appropriate answers. In this scenario, half the daughters are likely to be carriers, as I've just explained, and half the sons are likely to be symptomatic. All the other stems are incorrect. Question 3. In this situation, we have a couple who are both affected by the dwarfing condition achondroplasia. And in this situation, they want to know the chance of having a child who is of normal or average height. Now, people with achondroplasia do commonly come to genetics clinics, often to ask about the probability of a child being affected by homozygous achondroplasia, a lethal skeletal dysplasia, where the child inherits 2 copies of the achondroplasia mutation. But this is not what's being asked in this situation. In this situation, they're asking about the chance of having a child of normal, or what might be called average height. So how can we approach this question? We know that achondroplasia is an autosomal dominant condition. It's caused by, in most cases, one particular specific activating mutation in the FGFR3 gene. As I've said, embryos that inherit 2 copies of this mutation have a lethal dysplasia, and typically stillborn. But this is not the question that's been asked at this stage. So, there are essentially 3 ways to approach this question. All of them are of course right, but it really depends on how you prefer to think about the problem. You can approach it mathematically by asking what is the chance of the child being neither dwarfed nor homozygous, or you can think about it more pictorially by looking at the combination of zygotes, or you can reverse the orientation of the question and think of normal height as being a recessive trait that these 2 individuals carry. So let's look at each of those possible ways of approaching the question. The first way of approaching it is to say, is this a situation where we can do some calculations on the probabilities? Yes, it is. what is the chance of the individual being neither dwarfed nor homozygous? Now, we're joining probabilities here, and in this situation, these are mutually exclusive conditions. The child cannot be both normally achondroplastic and homozygous. They can be one or the other. The probability in this scenario of them being normal achondroplasia, a dwarfed individual, is 1 in 2. The probability of them having the lethal homozygous form is 1 in 4. And so the probability of them having neither is the sum of those probabilities taken away from 1. Adding those probabilities together gives 3 out of 4, and taking 3 out of 4 away from 1, of course, leaves you with 1/4. So that's one way of approaching it. The next way of approaching it is to think about how the zygotes are actually formed. In this situation, we have two individuals who each carry a copy of the mutated FGFR3 gene. In this instance, I've indicated the mutations as small a, and the normal wild-type gene as the big A. So this couple, each are affected by dominant achondroplasia, and it's possible that they might produce 4 possible types of offspring. Achondroplasia offspring, 2 chances out of 4. The lethal homozygous form of achondroplasia, 1 chance out of 4. Or 2 normal genes, big A, leading to a tall or what might be called average height individual, so 1 chance out of 4. The final way of looking at it is to simply reverse the orientation of the question, and to imagine that in this scenario we have 2 individuals for whom normal or average height is a recessive trait, and that each of them is a carrier for this recessive trait of normal height. In which case, we have 2 carriers for a recessive trait, and the probability of them having a child affected by this recessive trait is 1 in 4. So the answer, however you look at it, is 1 in 4. And really the way you approach the question is a matter of personal preference. Question 4 is another clinical scenario which we come across from time to time. In this situation, there's a condition called Tay-Sachs disease, which is particularly common in one ethnic population, the Jewish population. So a Jewish couple, Mr. and Mrs. Cohen, come to see you, and you're told there's a family history of Tay-Sachs disease. The family history is that Mrs. Cohen's brother had the condition. Mr. Cohen, as far as you know, is unrelated and has no family history. And you're told in the question that the carrier frequency in the population of interest in this population is about 1 in 25, and that there's no carrier testing DNA test results for this couple. So the question is, what is the chance that they will have an affected child? So how do we approach this? Think about it now, and write down the answer that you think is the correct one. So, we know that Tay-Sachs is a recessive condition, and we know that Mr. Cohen has a carrier population risk, which we've been told is 1 in 25. Mrs. Cohen is the sibling of an affected individual with a recessive condition, which means that both of her parents must be carriers, but Mrs. Cohen, because she's a healthy adult, is clearly not affected. Therefore, her risk is 2 out of 3. So we know the risks of both parents being carriers for the condition. So how do we use this information to tell us the risk of a child being affected? Well, we ask the question, how is it possible for a child to be affected in this situation? In order for a child to be affected by the condition, 3 independent events must happen simultaneously. Mr. Cohen must be a carrier, so must Mrs. Cohen, and even if they're both carriers, the child must inherit both mutations simultaneously. Each of those options, each of those requirements, has a probability as shown. They're independent events, and so the correct way to join these probabilities is to multiply them. And so multiplying them together, we get the risk of 1 in 150. Incidentally, you'll notice in the options you're being given here, option C, 1 in 37.5, is the result you get if you forgot to multiply by 1 in 4. And this is quite a common way of getting this question wrong, of simply calculating the probability of 2 carriers existing, but forgetting the probability that the child actually inherits the condition. Right, question 5. This is an interesting question. A girl comes to see you who's pregnant and eventually tells you, obviously reluctantly, that the child she's carrying was fathered by her paternal uncle. Now her concern is that her paternal uncle had previously had a child affected by Rett syndrome. So, she wants to know the probability that the child she's carrying would be affected by Rett syndrome. Write down your answer now. Now, what do we know about Rett syndrome? Well, we know it's one of the rare conditions which is transmitted as an X-linked dominant. It's due to mutations in the MECP2 gene and shows marked sex skewing. In fact, almost all the individuals who are affected are girls. However, the other thing that's very important to know about Rett syndrome is that the vast majority of cases are not actually inherited but are due to sporadic new mutations. So initially we can say that it's most unlikely that this girl would have a child affected by Rett syndrome because the vast majority of cases are due to new mutations. However, there are rare familial cases, but in the family history we've been given, we know that because this condition is only ever shown in girls, it's X-linked dominant, that the uncle, or the uncle's brother, that's the girl's father, could not be carrying the mutation, and therefore could not have passed it down to the girl. That means that we can reassure this young woman that the risk of her baby being affected by Rett syndrome is extremely low and will be population risk. Right, let's move on to question 6. Now, question 6 is about an X-linked condition, DMD, Duchenne muscular dystrophy. A couple come to see you, and the wife is pregnant. We know that her brother and her maternal uncle both had Duchenne muscular dystrophy. We've also been given some more information about this couple, which is that they've had a scan to show that the baby the lady is carrying is male. So in this scenario, what is the chance that this baby will be affected by DMD? Have a think about it, write down your answer now, and then we'll go through and see if you're right. Here's the family tree that we've just described. Mr. and Mrs. Brown and the baby of interest. Mrs. Brown, who has a brother and a maternal uncle affected by this X-linked condition. This tells us that Mrs. Brown's mother must be a carrier for Duchenne muscular dystrophy, because otherwise she wouldn't have been able to have an affected brother and son. If Mrs. Brown's mother is a carrier, then Mrs. Brown's mother would have 2 X chromosomes, as anyone would, one with a normal gene and one with a mutated gene. That means that Mrs. Brown has a 1 in 2 chance of having inherited the gene from her mother. So, if Mrs. Brown has a 1 in 2 chance of being a carrier for DMD, what is the chance that her son would receive that mutation? Because we know in this scenario that the baby she's carrying is male. Well, for her son to be affected, we have 2 independent events which must occur. One is that Mrs. Brown must be a carrier. We've said the probability of this is 1 chance in 2. The other is that even if she were a carrier, there's still only a 1 in 2 chance that the son would inherit the mutated X, because remember, Mrs. Brown, like all women, have 2 X chromosomes. One normal one, which she'll have inherited from her father, and one which she'll have inherited from her mother, and she will then pass on one or other of those X chromosomes to her son. So the chance of a son being affected is 1 in 2 times 1 in 2, or a total chance of 1 in 4. So the answer to this question is 1 chance in 4. Now this question is a little bit unusual because you've been told more information than you perhaps might have been in a different scenario. You've been told the sex of the child. Of course, if you hadn't been told the sex of the child, then the risk would be different because you'd have different information, and so your level of uncertainty would be different. If you didn't know the sex of the child, then you'd have to add in a third factor, that is the chance of the child being a boy, which of course is 1 in 2. But in this scenario you know that information, therefore it's not a probability, it's a certainty, and so you don't include it in the calculation. Now, we'll do a few extended matching questions now. So here we have a selection of possible answers to the questions that follow. The first scenario, A, is regarding a couple, Mr. and Mrs. Smith, who come along with a son who has Down syndrome. Unusually, you're told that Mrs. Smith has had a genetic analysis which shows that she carries an isochromosome for 21. And the couple are asking about the chance of their having another child affected with Down syndrome following the birth of their son Mark. So which of these figures do you think is the most likely estimate of their chance of having another child with Down syndrome? Make a choice now and write it down. Okay, this question rather hinges on whether you understand what an isochromosome is. So let me explain that if you're unclear. On the left you can see two photographs of chromosomes and a couple of cartoons to illustrate how they're put together. The small chromosome on the left is a chromosome 21, and you can see at the top it has a centromere, which is where the spindle attaches during meiosis to separate the chromosomes apart, and the rest of the chromosome below has been stained with Giemsa stain to allow you to identify which chromosome it is, and you can see it has a banding pattern which is characteristic of chromosome 21. And you can imagine that the genes on the chromosome 21 are arranged like the cartoon on the left, A, B, C. The chromosome photo on the right is an isochromosome 21, and essentially what this is two chromosome 21s stuck together end to end. And if you look carefully at the banding pattern of this isochromosome, you can see that it's a mirror image from top to bottom. If you look at the top and the bottom, you have a greyish band followed by a white band, then a dark band, then a white band, and then a thick dark band. So you can see, as in the cartoon, you have the mirror image of the chromosome top and bottom. So this is an isochromosome 21, which is effectively two chromosome 21s mechanically, physically stuck together, which means that at meiosis, when the chromosomes are supposed to separate into the two daughter cells, these two chromosome 21s physically cannot separate. They must both go into the same daughter cell together. That means, of course, that half the daughter cells will have two chromosome 21s in them, and half will have no chromosome 21. When these are fertilised, half the zygotes will be trisomy 21, and half will be monosomy 21. And as you may know, monosomy 21 is virtually never seen in clinical practice because this is a lethal outcome and would result in a miscarriage. So that means the only live-born results of this couple with the S chromosome 21 would be Down syndrome children. And therefore the probability of them having a Down syndrome child is 100%. They are certain to have it because there is no other way that those chromosomes can behave during meiosis. Scenario B. Another couple attend the clinic with their daughter, who has a clinical diagnosis of Noonan syndrome. In this situation, we are told that there's been no mutation found, and so this is a purely clinical diagnosis. However, we're also told that Mr. Jones, the father of Mary, you note has down slanting palpebral fissures, low-set ears, and has told you that he had surgery in the past for pulmonary stenosis. Now, hopefully you'll have some familiarity with Noonan syndrome and know that the characteristics we've been told about in Mr. Jones are features of this syndrome. And even if you're not that familiar with Noonan syndrome, this might be a reasonable guess for you to make in the exam. Also, you'll hopefully know that Noonan syndrome is inherited as an autosomal dominant condition. And here's a picture of an individual affected by Noonan syndrome. And you can see the down slanting palpebral fissures, the slits of the eyes that point downwards from the nasal bridge towards the temple, and the rather low-set ears, as well as the other facial features characteristic of Noonan syndrome. So in this situation, we have a dominant condition where the child and parent are affected, and so the risk of a future child being affected is 50%. The recurrence risk of any dominant condition where there's an affected parent. Scenario C. This is a case of cystic fibrosis. A couple attend whose daughter had cystic fibrosis. You're told in this case that you have got genetic analysis results, which shows that both parents are carriers of the commonest mutation found in the UK population, delta F508. So then, in this case, what is the risk of them having another affected child? Well, the genetic analysis information is actually a red herring. You don't need to know that at all, because this is simply a situation of the recurrence risk of a recessive condition. Both the parents have had a previous child affected by CF. As this is a recessive condition, the child must have two mutations in her genome, one from each parent. which of course means that both parents must be carriers. You can work that out just from the family tree. The genetic analysis has simply confirmed this. The practical advantage of this would of course be that in the clinic you would then be able to offer prenatal diagnosis should they choose to take that up. However, in this situation, all you need to know is that both parents are carriers for CF. So in that case, what is the risk of two carriers having a child affected by a recessive condition? Here's the scenario. We have an affected child which tells us both parents must be carriers, and the question is, what is the risk of the future child being affected? And the risk, hopefully you'll know, is 25%, or 1 chance in 4. Okay, so we've hopefully covered most of the common types of risk calculation questions you'll have in the exam. As I said, you need to have a basic understanding of what probability is and how you handle probability calculations. In order to understand and calculate risk in a genetic context, you need to know what the inheritance pattern of the condition of interest is, whether it's dominant, recessive, X-linked recessive, or, in a few cases, rarer inheritance patterns. You need to know the family structure, and it's often helpful to write out the family tree just at the side of the paper, just to make sure that you've understood what the relationships are and how the individual of interest fits in to that situation. Sometimes you're given extra information, such as the sex of the individual or other test results, in which case you need to think about what that does to the probabilities of the condition you're trying to work out. And then finally, make sure that you use the correct joint probabilities, you multiply or add, appropriate to the situation. Thank you.